Good morning, everyone. I'm Jun Ding from Stanford University, and it's my great pleasure to welcome you to today's lecture as a member of the program committee. Before we begin, please take a moment to silence all the electronic devices to avoid unwanted interruptions during the presentation. Also, a quick reminder about photography and recording policy. You may take photos of the lecture's work only if a green camera with a checkmark image is visible. In the absence of the logo, please refrain from taking pictures or videos or audio recordings. It is my great pleasure to introduce today's speaker, Dr. Takaki Komiyama, Professor of Neurobiology and Neurosciences at UC San Diego. His lab focuses on investigating how ensembles of neurons in behaving animals adjust their activity throughout learning and using advanced technologies such as two-photon calcium imaging combined with electrophysiology and genetics, behavior, and computational modeling. Dr. Komiyama earned his bachelor's degree in biochemistry and biophysics at the University of Tokyo, and his PhD in neurosciences from Stanford University in the lab of Li-Chun Luo. Followed that, he joined Dr. Carol Svoboda's lab at the Genetic Farm, did his brief postdoc where he helped pioneer in vivo two-photon calcium imaging in awake behaving animals. Since he joined UCSD, his work is especially significant to the neuroscience community because he has helped push forward on how we view learning and circuit plasticity at very different levels, from individual synapses, individual spines, to population of neuronal ensembles. For example, his group showed that how new dendritic spines form and become functionally integrated during acquisition of motor skills, thereby revealing synaptic and circuit levels changes that underlie long-term motor memory formation and adaptive behavior, which I think he will elaborate in more detail during his talk. These insights have really broadened our understanding of how learning reshapes brain circuits and how functional clusters of synapses emerge and how distributed networks of neurons reorganize to support learned behavior. His combination of imaging, genetic tools, computational approaches has really set a new standard for studying brain plasticity in behaving animals. So following Dr. Komiyama's talk, we'll hold a Q&A session, so please plan to stay until the end. And you may submit your questions at any time during the talk via the meeting apps or virtual platform, and I will collect as many of your questions and then post them at the end. So if you have to leave before the Q&A, please exit quietly from the rear of the room, so not to disrupt the presentation. And lastly, please know that the session is being recorded and will be available till December 21st. So with that, please join me welcoming Dr. Takaki Komiyama.
Thank you so much, Jen, and thank you, Program Committee, for this opportunity. I am very excited to share some of our latest discoveries with you all, so thank you all for tuning in. And I have to admit that I was not aware of the logos to tell you that you can record this, but I don't mind, so please take pictures, take videos as you wish. Be my guest. Making adaptive body movements is really one of the most fundamental functions of the brain. And in fact, it is the only way by which animals like us can act on the environment and our conspecifics. And as such, the brain dedicates a lot of its real estate to generate voluntary movements. And in this slide, I'm just highlighting just a few of the brain areas that are primarily implicated in movement control. And these interconnected areas have complementary functions, and they work together to control body movements in an adaptive manner. And in particular, my lab's research focuses largely on the primary cortex, or M1, and its associated circuits. And we believe that M1 has a specialized function in learned movements. And this function can be evident in tasks like this one. So this is the task that my lab has been using for the last 15 years now, in which head-fixed mice use one of its forelimbs to grasp a lever to move it to receive a reward. And it's a simple enough task that even on the first day of training, they can perform this task to some extent. But when you look at the movements on individual trials, you see that they are highly variable from trial to trial. However, with daily training over 2 weeks, they develop a more consistent pattern of movement that we can observe reproducibly across trials. And in fact, this consistency of movements is really a hallmark of well-learned motor skills. And extreme examples of these can be found in professional athletes. And I'm going to share an example from a professional baseball pitcher. This is this year's World Series MVP, Yoshinobu Yamamoto of LA Dodgers. And when I play this video, because its form is so consistent across pitches, you may not even realize at the beginning that it's actually 2 pitches overlaid on top of each other. And this consistency is really remarkable, so I'm just going to play that one more time. And I think it is so cool. I grew up playing baseball, and he is really living my dream. And even though this is a really simple task in head-fixed mice, we think that this captures this important essence of learned motor skills, this consistency of learned movements. And another important feature of this task is that it involves dexterity of distal muscles. So the lever is a thin rod that they have to use distal muscles to grasp, to manipulate. So it's different from just a wide lever that they can just smack on. And this importance of dexterity makes this task particularly dependent on M1. And in fact, when we lesion the primary motor cortex M1 contralateral to the used limb prior to the training, they can still learn the task, they can still perform it to some extent, but they never develop reproducible, consistent movement. Okay, so this learning of this consistent movement, it really depends on M1 functions. And this is not because M1 is required for plasticity elsewhere, but instead, plasticity within M1 we think is critical. So for instance, in our recent experiments, we used an optogenetic tool, optogenetic inhibitor of CAM-K2, and we expressed this locally within contralateral M1 to block local synaptic plasticity in M1. And this manipulation was sufficient to reproduce the lesion phenotype. So compared to the control animals that develop more consistent movements, when M1 plasticity is blocked, they never develop the consistent movement. And this is despite the fact that the rest of task learning is intact, and they can achieve a reward in a comparable fraction of trials. So they know, they learn that lever movement leads to reward. So that type of learning is not dependent on M1, but the learning of the consistent movements is dependent on M1. So building on this framework, I will share with you some of our latest results studying cortical circuits for learned movements. I will start by describing the organization and function of M1 circuits for learned movements. And then I will discuss the mechanisms involved in how this learned circuit forms during learning. And then I will end by describing our latest efforts trying to leverage some of the mechanisms that we learned from studying healthy animals to potentially treat a disease. To begin to study the organization and function of the circuits for learned movements, we applied 2-photon calcium imaging in M1 longitudinally to record the activity of the same population of M1 neurons throughout learning, to ask whether the activity of M1 might change during this learning. And I'll summarize the main finding in this slide. So here, each color represents the activity timing of a given neuron across trials. And hopefully what you can see is that the activity timing of each neuron is highly variable from trial to trial at this beginner stage. But with daily training, each neuron picks a favorite time to be active, so that the population activity becomes refined and a consistent sequence of activity is observed across trials. So now the interpretation would be that M1 learns the ability to generate this activity pattern consistently, and that is causally driving the consistent execution of the learned movement. But there's always this chicken and egg problem, which is very difficult to address in studies of movement encoding. Is this activity pattern really driving the behavior, or is this a result of the movement, perhaps as sensory feedback? And we began to address this question using 2-photon optogenetics, which allows us to stimulate essentially any group of neurons within the field of view. And we asked whether we can stimulate these neurons, these early onset movement neurons whose activity onset precedes the beginning of voluntary movements, to see if that stimulation can induce some of the learned movements. And we do this in the task context. So most of the trials are voluntary trials. Mice are waiting for the go cue to press the lever to get the reward. But in a small fraction of catch trials, we omit the sound cue, so mice wouldn't press the lever voluntarily, but instead we stimulate 20 of these early onset movement neurons to see what happens to the behavior and the circuit dynamics. And I'll start by showing you two videos. Okay? On the left is one of the voluntary trials where you see the left limb grasping onto this thin lever. I'll play the same video a couple of times, okay? Just to highlight this left forelimb grasping the lever, and then there's the, oops. Okay, press and hold, and then return. Just playing it one more time. Press and hold, and return. So hopefully you can appreciate that, even though this is a simple task, the movement itself is far more complex than just simple twitches. And on the right now, I'm going to show you a video from one of the opto trials where we observe really qualitatively similar movements, starting from the press and hold, and then return. So when I first saw this, I was really surprised. I didn't necessarily expect that stimulation of just 20 neurons in M1 could drive the entire sequence of the learned movement. And the assumption is that we don't think that 20 cells alone are sufficient for such complex behavior, but stimulation of 20 cells, their activity spread to the rest of the network, and this population activity together collectively drives the learned movement. And in fact, when we so to test that, we looked at the activity of the non-target neurons. So these are the neurons that were not directly stimulating. And in voluntary trials, we see cell-specific activity. So a fraction of neurons show activation during voluntary movements. A small fraction are suppressed, and other neurons are not modulated. And a very similar activity pattern is reproduced in the opto trials. So again, these are not the cells we're directly stimulating. We're just stimulating 20 cells, but that is enough to drive this population activity pattern that is very similar to the learned activity pattern. So it appears that the M1 has a pattern completion function, and the stimulation can lead to the completion of the learned activity pattern. And at that point, we were wondering, maybe this pattern completion function is so strong that any random stimulation can evoke the learned activity pattern. So to address this, we stimulated intentionally the wrong cells. So these cells, late onset movement cells, whose activity onset lags the beginning of voluntary movements. And so that when we stimulate 20 of them, we don't induce movements in most trials. And in the small fraction of trials where we see movements induced, those movements do not resemble the learned movement. So to reliably drive the learned movement, we need to stimulate the right cells, namely those early onset cells. So our interpretation is this. So M1 has an intrinsic circuit that can pattern complete this learned activity pattern. And when this circuit receives the appropriate input that stimulate the first starter cells, then that activity spreads to their neighbors to complete the learned activity pattern. And that induced activity pattern would consistently drive the learned movement. But even at this point, there's still part of the chicken and egg problem that remains. And one can come up, in theory, different scenarios. So in this alternative possibility, we're stimulating 20 cells. And those 20 cells are just directly driving the learned movement by engaging downstream circuits. And maybe what we see in the rest of M1 is all still just a result of the learned movement, perhaps as sensory feedback. And again, this chicken and egg problem is really difficult to address in studies of movement encoding. But our stimulation experiments give us a unique leverage to address this. So far, I've shown you that stimulation of those early onset neurons can induce the movement and see the induce the activity pattern in M1 that resembles the learned activity pattern. However, the stimulation doesn't always induce movements. So just in about 60% of the trials, we see movements induced. And in the remaining 40% of trials, the same stimulation does not lead to overt movements. And strikingly, in those 40% of no movement trials, we still see M1 dynamics that somewhat resemble the learned activity pattern. Okay, so this may not be quite as good as what we see in movement trials, but still, the pattern of activity is substantially similar to the learned pattern than expected by chance. So M1 dynamics can be induced independent of movements. And based on this result, I feel comfortable rejecting this alternative possibility. The main difference between these scenarios is that M1, in the top working model, M1 plays an active role in generating this learned activity dynamics, while in the alternative possibility, M1 simply reflects passively the incoming information. And we conclude that M1 indeed plays an active role in generating this learned activity pattern independent of movements. And this pattern completion function of M1 is state dependent. So far, all of our stimulation experiments are done when the animal is engaged in this task, preparing the movement. When the same experiment is done, stimulating the same cells in the same animals in a no-task context, when the animal is not engaged in the task, the same stimulation fails to induce any movements or the learned activity pattern. Okay. So this pattern completion circuit, pattern completion function in M1 requires that the animal is engaged in the task preparing the movement. So this M1 intrinsic circuit that can complete the pattern becomes primed when the animal is engaged and preparing the movement. And in this primed state, the stimulation of those starter cells can lead to the complete pattern driving the learned movement. And so far, I've been talking about how this can be induced with artificial stimulation of the starter cells. But of course, that's not how the animals generate voluntary movements. So we wanted to know where the input comes from within the brain when the animal voluntarily generates the learned movement. And we addressed this issue by casting a wide net, basically recording all of the inputs to M1 from outside of M1. And these inputs come from motor thalamus, primary somatosensory cortex, contralateral motor cortex, and premotor cortex. And we examined all of these inputs one by one by expressing GCamp in those input areas and imaging their axon terminal activity within M1. And we find that these areas bring different information to M1. And in particular, inputs from the thalamus were unique in that their population average activity began to rise before the movement starts. Okay? So they were the prime candidate for driving the starter cells to initiate the dynamics. So the hypothesis is that thalamic inputs selectively activate these starter cells, and then M1 spreads that activity to the rest of the network to complete the learned activity pattern. So to test that hypothesis, we wanted to identify the neurons within M1 that are activated by the thalamic inputs. And we did this by expressing an excitatory opsin in the motor thalamus and activating their axon terminals in M1 while we record the activity of M1 neurons. And this approach allowed us to identify a small fraction of M1 neurons that are consistently excited by thalamic inputs, these thalamus-excited neurons, that were intermingled with a large majority that were not strongly driven by thalamic inputs, these thalamus non-responsive neurons. So with this ability to identify the small fraction of thalamus-excited neurons in the sea of thalamus non-responsive neurons, we then went on to record their activity to characterize their functional properties during the voluntary movements and find that, indeed, thalamus-excited neurons are highly engaged during the learned movement, with their activity beginning prior to the movement onset. So some of these are those early onset neurons. So thalamus is indeed activating those starter cells. Interestingly, this is a function that seems to evolve during learning. And at the beginner stage, this effect is not very clear. So somehow this thalamocortical influence becomes refined during learning, and thalamus acquires the ability to reliably and strongly activate those starter cells in M1. Consistent with this idea, when we inactivate the thalamus using chemogenetics, in expert animals, we see a major impairment in the execution of the learned movements. They even often struggle even initiating any movement, indicating that the execution of the learned movement requires thalamic activity. And consistent with the idea that this thalamocortical pathway is something that becomes engaged with learning, strikingly, the same manipulation at the beginner stage does not have a major impact on the ongoing behavior. So taking these together, we think that there are two parallel changes that occur during learning. Within M1, the pattern completion circuit forms that might look like this through local plasticity of local recurrent connectivity. And at the same time, the thalamocortical pathway becomes refined so that the thalamus learns to activate the correct starter cells. After these circuits form, when the animal prepares the movement, it engages this pattern completion circuit in M1. And in this primed state, thalamus activates the starter cells, and then the rest pattern completes autonomously to drive the learned movement consistently. So now we're going to ask, what is the mechanism that allows the circuit to refine this way? So how does the learned circuit form? And a lot of us assume that this type of learning involves synaptic plasticity. And in fact, early on, I showed you that when we block M1 plasticity locally, then the behavioral learning is impaired. And when we image the activity of M1 neurons in this same context, we find the following. So in control animals, the activity timing of individual neurons becomes more consistent across trials, and the population activity sequence becomes more refined. And this refinement of M1 activity is also impaired when we block local synaptic plasticity. And this is significant because a lot of times, when we talk about refinement of M1 activity, people tend to assume it's the refinement of input activity. But this result shows that actually local plasticity contributes significantly to this activity refinement. All right, so synaptic plasticity is involved within M1. But for synaptic plasticity to generate such a specific circuit, the plasticity needs to be regulated very precisely. The plasticity needs to modify the right synapses at the right time. And in this part of my talk, I will describe some of the mechanisms that we've identified that ensure this spatial-temporal specificity of plasticity. And I'm going to start with the temporal specificity mechanism. And for that, we focused on this disinhibitory circuit in the cortex with the VIP inhibitory neurons at the upstream of this, at the top of the hierarchy. So VIP neurons are special inhibitory neurons that mainly inhibit other inhibitory neurons. And when they become active, they inhibit downstream inhibitory neurons, leading to the disinhibition of excitatory neurons, allowing them to become more plastic. And we found that when an animal is trying to learn a new movement, VIP neurons are strongly activated during those movements, engaging this disinhibitory circuit. And we found that this disinhibition is critical to open the temporal window of plasticity. So essentially, it creates a permissive environment just transiently for plasticity to occur so that the circuit can reorganize and learn the movement. And when we interfere with this process, perhaps, for example, by activating one of the downstream inhibitory neurons, somatostatin inhibitory neurons, then we can block the learning-related synaptogenesis, and this also blocks the ability of the animal to learn the consistent movement. So this transient disinhibition is a critical mechanism that allows the window of plasticity to open transiently, specifying the temporal specificity of plasticity. And this is a circuit that we will come back to at the end of my talk, so keep that in the back of your mind. And now we're going to shift to the spatial specificity of of plasticity. So even though this is one of the mechanisms that defines the temporal specificity, we still need to modify the right synapses. And in fact, learning-related plasticity is synapse-specific. So here's an example where we are imaging the same group of synapses across days. And we can identify some synapses that become larger, corresponding to their potentiation. And their neighboring synapses can become smaller, corresponding to their depression. Well, the question is, what about this synapse made them to become stronger? And what about this synapse made them to become weaker? To begin to address this question, we wanted to measure the types of information that are available to these synapses. And the main information that is available to these synapses is the activity of pre- and postsynaptic neurons. And so we established a method to measure both of these simultaneously with this dual color imaging with two sensors, one for glutamate, which measures the release of glutamate onto individual synapses, And the other sensor is for calcium that reports the postsynaptic calcium activity. And so the data look like this. In blue, you see glutamate release onto individual synapses that are simultaneously imaged. And in red, you see calcium activity in dendrites, with the goal of identifying the patterns of activity that predict the plasticity of individual synapses. And we considered the fact that the neurons have a complex morphology, and different dendritic compartments have different computational functions. Apical dendrites are enriched with synapses receiving long-range inputs from outside of M1, while basal dendrites are enriched with connections from nearby neurons, the local recurrent connectivity. And I'm going to summarize the main findings in this slide. In the apical dendrites, synapses that become stronger tend to receive correlated inputs with nearby synapses. In other words, when glutamate is released onto neighboring synapses in a correlated manner, those synapses tend to become stronger. And this cooperation of neighboring synapses is completely absent when we looked at basal dendrites. But instead, in the basal dendrites, we find that those that become stronger tend to receive glutamate at the same time as the postsynaptic neurons' activity. And this is exactly what you expect from Hebbian plasticity that says neurons that fire together wire together. And that's exactly what we're seeing here. When the pre- and postsynaptic neurons are coactive, the connection between them becomes stronger. And this evidence for Hebbian plasticity is completely lacking when we looked at apical dendrites. So there's a really nice double dissociation here. Neighboring synapses cooperating with each other through coactivity in apical, but not basal synapses, while Hebbian plasticity is observed in basal, but not apical synapses. A prediction of these observations is that the basal plasticity is fundamentally dependent on postsynaptic action potentials, while the apical plasticity may not be. And we tested this prediction directly by using the KIR potassium channels, which can cell-autonomously inhibit action potential firing. So here you see that in magenta, the sparse group of KIR expressing neurons that fail to fire action potentials, surrounded by normal KIR non-expressing neurons. And using this system, we compared the plasticity of silent neurons surrounded by other normally firing neurons. And strikingly, this manipulation really did not affect much about apical plasticity at all. So in these apical dendrites, even though the postsynaptic neurons are silent, these synapses have no problem undergoing LTP and LTD. However, the picture was very different when we looked at the basal dendrites, where potentiation was almost completely abolished with a corresponding twofold increase in LTD, indicating that postsynaptic action potentials are critically important for basal plasticity, while apical plasticity is independent of it. So we started by asking, what is the plasticity rule that determines the synapse specificity of learning-related plasticity? And we learned that there are actually multiple rules, even within individual neurons, depending on the subcellular location of the synapses. And apical plasticity is based on coactivity of neighboring synapses, and this mechanism would favor the formation of functional clusters of correlated inputs, while basal Hebbian plasticity promotes the formation of Hebbian ensembles locally. And even though I can't say that I necessarily predicted these findings, but they still fit very nicely in hindsight into the framework of this learned circuit. Specifically, the long-range inputs from the thalamus would arrive in apical dendrites, and through the cooperation of neighboring synapses, this plasticity rule favors the formation of functional clusters of long-range inputs. And these functional clusters, when they're coactive, can take advantage of the supralinear integration mechanisms of dendrites to strongly drive the postsynaptic neurons. And this can happen across multiple neurons. And when these neurons are coactive, basal Hebbian plasticity rule would bind them together to form Hebbian ensembles that have the pattern completion function. And so this interplay between long-range inputs forming functional clusters with local connectivity forming Hebbian ensembles for pattern completion would work together to consistently drive the learned movement. And now I'm going to switch gears a little bit, and I'd like to use the last 10, 12 minutes of my talk to describe some of our latest efforts trying to leverage some of the plasticity mechanisms that we've identified in healthy animals to try to treat a disease. And for this, we will focus on this disinhibitory circuit. Just as a reminder, I told you that this is a circuit that becomes engaged when an animal is trying to learn. Yeah, so when the animal is trying to learn, that activates VIP neurons, leading to the disinhibition of excitatory neurons to transiently open the window of plasticity. And this is the circuit that we study in the context of Huntington's disease. So as many of you know, Huntington's disease is a devastating, progressive, and fatal movement disorder. And the genetic cause of this disease is very well understood. It's a mutation in a single gene, Huntingtin. And when you have that mutation, you develop the disease without exception. And despite this very well-defined genetic cause, there's currently no cure or effective treatment. And thanks to this single gene nature of the disease, it is relatively straightforward to model in animals. And in fact, when this human mutation is introduced into mice, then those mice develop Huntington's-like phenotypes. So we're going to use those mouse models and study cortical circuits. And for aficionados out there, I'll show you results from the transgenic R62 line. But we also reproduced the main findings using another mouse line, the knock-in ZQ175 model as well. And we studied this circuit in Huntington's models using this task, where head-fixed mice are placed on a ladder that is motorized. And we rotate the ladder with variable intervals, so the mice need to grasp the rungs of the ladder and walk on it to keep up with the rotation of the ladder. And this skilled locomotion task is convenient because we do not need to water restrict the mice, which is difficult to do in disease models. And in using this task, we're going to investigate cortical circuits in a cell type specific manner, starting with the VIP neurons that are at the top of this hierarchy of this disinhibitory circuit. When we image the activity of VIP neurons in both control and Huntington's models, we see a dramatic phenotype. So in blue, it shows movement-related activity of VIP neurons in wild-type animals. So as I said earlier, when an animal is trying to learn a movement, these neurons become highly engaged. And that's what we see in wild-type animals. And this movement-related activity is completely missing in Huntington's mice. Okay? And this is not a subtle phenotype. It's really a consistent population activity that we see in every animal that is completely missing in disease models. And this is not because the cortex is generally hypoactive, but it's cell type specific. So when we look at their downstream inhibitory neurons, like the somatostatin inhibitory neurons, we see the opposite phenotype, where the movement-related activity of somatostatin neurons becomes strongly exaggerated, as shown in this red line here. Same is true for PV inhibitory neurons. Also, the movement-related activity is strongly exaggerated in the disease mice. And so these inhibitory neurons are hyperactive, which would result in hyperinhibition of excitatory neurons. And consistent with that idea, when we look at the activity of excitatory neurons, we see that they are hypoactive. Their movement-related activity is strongly reduced in the disease. So we see a very clear cell-type-specific phenotype in this disease model mice, with VIP neurons showing hypoactivity and perhaps downstream circuits showing stronger activity in other inhibitory neurons, leading to hyperinhibition and lower activity of excitatory neurons. And stepping back a little bit, this is the mechanism that I showed you earlier as essential for normal learning and function of cortical circuits. Okay, so when an animal is trying to learn, VIP activity kicks in to open the plasticity window. And this circuit seems permanently hypoactive, underengaged. And so that the cortical circuits seem to be stuck in the non-plastic state. This would be a problem, and can we fix this? And of course, that's the ultimate goal. And because these cell types are genetically targetable, it is straightforward to try to repair or normalize this cell type-specific dysfunction. And we turn to the stimulation of VIP neurons because they are at the top of the hierarchy of this inhibitory circuit. So we used optogenetics to stimulate VIP bilaterally in M1 during movements of the latter task in 40% of trials. And we did this repeatedly across 9 different sessions, okay, over a couple weeks. And we first wanted to monitor what this does to the network's activity. First, by looking at the activity of VIP neurons. So these are the neurons that we're directly stimulating. And we find that in the stimulated animals, we can restore the healthy level of activity of VIP neurons. So down here is the activity of VIP neurons in disease models without light stimulation. And in the trials with light stimulation, we can restore their activity to healthy levels. And this can be done across the experiment throughout 9 sessions. And this is not surprising. This is exactly what you would expect. So we're stimulating these neurons, and we're confirming that our stimulation is selectively and normalizing VIP activity. Our optogenetics is working reliably. What's even more exciting is seen in downstream neurons. So when we look at the activity of excitatory neurons, early on in this manipulation, we see this disinhibition expected from stimulation of VIP neurons. So their activity is slightly enhanced during the light trials. However, when we do this repeatedly across 9 days, we begin to see that their activity becomes generally more enhanced, even in the no-light trials. Okay? So in this dark red line, we're not applying the light in those trials, but their activity is closer to what we see in healthy animals, much higher than what we see in other animals, other disease animals without stimulation. Okay. So this repeated stimulation of VIP seems to be engaging the endogenous plasticity mechanisms to enhance endogenous plasticity, which allows the circuit to repair itself so that eventually the circuit gains the ability to express more normal activity levels independent of the acute stimulation. And of course, finally, what we would finally want to see is that this would help behavior, and we are very happy to report that it does. So here I'm going to show you one of the behavioral measures that we use, hind limb dragging. So when the animal has trouble keeping up with the movements of the ladder, they start dragging the hind limbs. And in control animals, we see little of this, and this even improves with repeated training, indicating motor learning. And in disease mice, this gets worse and worse over time, reflecting their disease progression. And we can reverse this worsening of behavior by VIP stimulation, repeated VIP stimulation. And close, not quite, but close to the levels that we see in healthy animals. And what I'm most excited about is that this behavioral effect persists beyond the stimulation. So after this 9 days of stimulation, we performed 2 additional sessions where we're not stimulating VIP neurons. So sessions 10 and 11 corresponding to 2 and 4 days after the last stimulation session. And this behavioral improvement persisted in those no stimulation sessions, indicating that our VIP stimulation caused a lasting improvement of their behavioral performance. And this hind limb dragging is a really specific measure of their behavior, but we see this improvement across various behavioral measures. So their cadence becomes faster, close to the level that we see in healthy animals, and their gait becomes more regular, and the trajectory of their movements become more direct and straight. So these are all, thanks to VIP stimulation, we see the improvement in their performance in this skilled locomotion task across the board, across different behavioral measures. Thus, by opening the window of plasticity through VIP stimulation, we were able to provide persistent improvements in the skilled locomotion task. And we propose this as a potentially generalizable intervention to re-engage cortical plasticity to repair diseased circuits to provide lasting behavioral improvements. And this is an exciting full circle moment for us. In the first parts of my talk, I shared with you some small slices of our 15-year endeavor scrutinizing the basic neurobiology of motor learning. And we have been investigating this at various scales, from synapses to cortical microcircuits to long-range pathways, really trying to understand the detailed mechanisms by which the brain learns to move. And of course, understanding how the brain works is inherently satisfying. I don't need to tell you that to this crowd. But also, I have been increasingly realizing that, borrowing the words of the baseball great Jackie Robinson, neuroscience is not important except in the impact it has on society. And of course, one of the ways by which neuroscience can impact society is by improving the quality of life of patients. And that is why it is personally satisfying that our research is finally coming to a point where it uncovered at least one point of leverage that we can take advantage of to potentially treat a disease. And at least in the mouse models of Huntington's disease, this manipulation is allowing us to devise a treatment that can provide lasting improvements in their behavior. And of course, we do not believe that our treatment is fixing everything underlying disease phenotypes, so we plan to go back to these detailed measurements to try to identify what is improved and what is still to be improved, so that we can design additional treatment options to further improve these disease circuits. So it is a very exciting time for us. And I end by emphasizing that I did not do any of these by myself, and rather it is really the fruit of many, many individuals, dedicated scientists who go through my lab, as well as my collaborators. And it has been a real pleasure to be able to work with them and share this journey with them. And thank you all for your attention.
Thank you, Takaki, for the wonderful talk. There are many questions that are still rolling in on the neuroscience app and virtual platform. I'll try to raise the questions, especially the ones that got asked repeatedly. So the first question is about what's the physiological process in the cortical circuits causing slips in learned motor behavior? For example, professional ball players have wrong pitches or missed ball catches.
Yeah, excellent question. So this is about choking under pressure. And this might have to do with something that we've studied but didn't have time to study, time to talk about in this talk. So we have found that in highly learned context, the learned skills can become independent of M1. So this function might become offloaded to outside of the cortex. And we suspect, we speculate that this happens for a more automatic and almost like non-cognitive execution of highly learned skills. And one of the theories that have been thrown around about choking under pressure is that when the skill is so highly learned that it is largely subcortical, the cortical activity can add noise to their functions and perhaps mess with the reliable execution of the movement by downstream subcortical circuits. But that's something that we didn't talk about today, so I appreciate the question.
Yeah, thank you. So the next two questions are related and a little bit detailed. So you showed that stimulation of 20 neurons can drive M1 to complete the pattern. So what happens if you stimulate 19 or 18? In other words, is there any minimum number required for this pattern completion? And then related, when you inhibit these 20 cells, what happens?
Yeah, excellent questions. So to address the first question, do we need to stimulate 20 cells? The first approximation, yes. So we've done a limited amount of experiments looking at the result of stimulation of 5 and 10 neurons. And those stimulation sessions, stimulations lead to a less reliable induction of movements. So we get movements in a smaller fraction of trials. And the induced movements do not resemble the learned movement as much as when we stimulate 20 cells. So 20 cells is close to the stimulation threshold, at least in our stimulation thresholds, stimulation parameters. And what happens when we inactivate these cells? So we haven't been able to do that. My understanding is that still targeted inactivation is difficult. But so I'll only speculate here. So the reason we think that the 20-cell stimulation works is not because 20 cells is really sufficient, but it's also because the intrinsic circuit in M1 is able to complete the pattern. So our stimulation spreads to other neurons within the heavier ensembles to amplify the stimulation to complete the pattern. So within that framework, I suspect the inactivation of those neurons may not be enough to drive a strong behavioral impairment. So the rest of the circuit might still be sufficient to generate the activity and movement. But that's something that needs to be tested experimentally.
Yeah, next question again is related to the pattern completion. So how does pattern completion develop over time as a complex learned arm movement is executed over the period of time?
Okay, so by observing the activity in voluntary learning trials, we see that the activity becomes more and more consistent. That suggests that plasticity during this learning is reorganizing the circuit to generate a pattern completion circuit. So early on in learning, we speculate that the same experiment, for example, stimulation would not lead to the completion of any pattern. So this is before the pattern completion circuit has formed. But to directly test that idea, there's a lot of drift in the activity patterns, so we would have to invent a time machine to identify the key cells at the end of learning and go back in time and stimulate those cells at the beginning, which we're still working on making the time machine, but it might take a little longer.
Well, next one probably is related to time machine again. So if what happens when the mice are learning a different task? Do they require the same population of the starter cells?
Yeah, that's exactly what what we would like to study more and more. In a previous study, we examined M1 activity when the animal is alternating different movements, two different forelimb movements. And we saw that activity related to one movement and the other, they were partially overlapping. So some cells participated in the ensemble for both, but many cells were unique to one or the other movement. So we suspect that these pattern completion circuits can coexist for different movements. And M1 must have the ability to engage or prime one and not the other on a moment-by-moment basis. But that's something that we'd really like to study in detail using our stimulation paradigms. So stay tuned.
Yeah. So given what you described about the functional distinction between the apical and the basal dendrites, how would you speculate on the impact of sleep promoting the plasticity?
Yeah, absolutely. So that's another thing that I've said for 15 years that my lab will get into, and we still haven't gotten to that. Previously, when we were looking at synaptogenesis during learning, a lot of synaptogenesis happened during the sleep, or at least overnight. Okay. So and there are other studies, not from our group, but the other studies suggesting that M1 has the ability to replay some of the movement-related activity during sleep, which might be important for consolidation of learning-related plasticity. So yeah, sleep is important. A lot of the plasticity is likely enforced during sleep. My lab has not looked at that in detail.
So what are the possible reasons leading to that about 30 to 40% of trials during M1 stimulation that do not lead to expected movement?
Yeah, I appreciate this question. This is something that I decided to cut off from the talk just for the sake of time. So I showed you that in 40% of the trials, stimulation does not lead to the movement. And there's additional variability that I did not have time to talk about. So in the 60% of the trials, when stimulation leads to a movement, the movements are also variable. In some trials, we get this really nice learned movement, like what I showed you in the video. But in other trials, we still get some movements, but not the perfect learned movement. So there's a lot of variability despite the same stimulation that we are applying across trials. We're still exploring what determines this variability. One of the observations that can predict this variability is local activity in M1. So looking at the activity half a second before we stimulate these 20 cells, we can predict if it's going to be a movement trial and if it's going to be the good movement trial or bad movement trial. So this suggests that there are different levels of movement preparation reflected in the different levels of priming of the M1 circuit, and reliable induction of the learned movement really depends on the proper initial state of the network. So that's at least one of the things that can predict the outcome of our stimulation.
Cool. So the next set of questions are regarding to the manipulation of the circuits for therapeutic treatments and improvement of the motor behavior. So is there any promise in artificially opening the temporal window of plasticity to accelerate or to refine the motor learning?
In healthy animals, I think,
yes,
right?
Exactly.
Yeah, of course. We all want to make ourselves smarter. And There's a bit of a hint here. So if you compare this light blue and dark blue lines, so light blue is wild-type animals with VIP stimulation, and dark blue is without. And maybe we're making the wild-type animals a little better by stimulation of VIP neurons too. So, but of course, the effect is much smaller. So in the disease animals, the behavior is so bad that there's a lot of room for improvement. We suspect that healthy animals have the system tuned so that it's near optimal, so it's difficult to further improve. But maybe we're seeing a little evidence of that. But what we would really like to do in the future is try to treat healthy individuals, but in a somewhat compromised state. And one thing that we all humans do is to age. And we all know that our learning abilities become blunted with aging. And perhaps, I'm just spitballing here, speculating, but this is effective intervention to help aged brains learn better. So that's something that we'd like to explore in the future.
The last question, given the time, is going to focus on the Huntington's disease model. So in your HD disease models, when you stimulate VIP, you improve the behavior. How does that impact on neurodegeneration? Does that protect the neurons from dying?
Yeah, excellent question. So that's one of the first questions that we asked when we saw these behavioral improvements. Long story short, we don't know yet, and we're going to look at them. But I'm almost certain that this is not halting neurodegeneration, right? Maybe there's a hope that some of the degeneration might be slowed a little bit thanks to normalized activity patterns. But yeah, I'm almost certain that neurodegeneration is still ongoing. So we propose that our treatment is something that should ideally be combined with more molecularly targeted treatments to directly target either the broken gene or broken proteins to slow the neurotoxicity and neurodegeneration. So combination therapy might be even more promising.
All right, thank you very much. And
thank you.
Let's thank Dr. Komiyama for a wonderful lecture, and thank you all for staying.
Thank you very much.